Perfect. So we're at noon, so I just want to say hello to everyone that's joining us for this talk today. So this is our Cryo-EM current practices webinar. And so we are recording, and you will be able to find today's talk along with our previous talk on our YouTube page or on our events page at cryoemcenters.org. So you're joining the 3 national centers that have been established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Lauren Vega, and I am the project coordinator for Pacific Northwest Cryo-EM Center, or PNCC. And then with me today are Ed Ang and Eugene Chua from the National Center for Cryo-EM Access and Training, NCCAT, as well as Michael Schmidt from the Stanford Slack Cryo-EM Center, S2C2, and then today's speaker, Liz Kellogg, who was invited by NCCAT. So this webinar series is hosted regularly on the last Thursday of every month, focusing on cryo-EM. Along with exciting biology, these talks highlight the methods and research groups that research groups use to obtain and interpret cryo-EM data that they collect with our National Center resources. So we do hope that these talks are useful, especially for groups starting out in cryo-EM. So Eugene will actually introduce Dr. Kellogg properly after each of the national centers gives a quick update on the cryo-EM resources they have available at no cost to the cryo-EM research community. And then regarding the next talk, on March 31st, PNCC will actually be hosting Do-Hoon Kwon, who is a postdoctoral scholar with Dr. Lee at Duke University, who will tell us their work in heat-dependent opening of TRPV1 in the presence of capsaicin. We ask that you use the Q&A button to send questions at any time during the talk, or upvote questions you will see there. Eugene will moderate the questions at the end of the talk. And with that, I'm going to turn it over to our center representatives. So Michael, you can start us off.
Hi, I'm Mike Schmidt. I'm one of the co-directors of the Stanford Slack Cryo-EM Center, S2C2. And as for the other 2 centers, as with the other 2 centers, we have Krios microscopes with the latest detection equipment on them and energy filters. We have projects for data collection, for training, and for screening at our center, like the other centers do. And we were just there at the Biophysical Society meeting. If you pass by, you could see us at a booth there. We'd like, if in August when you come to the my M&M meeting in Portland, we'll have a presence there along with the other 2 centers. Come by and see us, and at any time you can contact us at this website. Thanks a lot.
Thank you, Mike. So I'm going to go next for PNCC. So we now offer 2 proposal types. So we have a screening access for up to 5 days of sample preparation optimization targeted to low resource institutions. Then we have our general access for single particle analysis or tomography, which gives up to 480 hours a year and valid for 2 years. So approved general access proposals are eligible for our training workshops. And we have a monthly submission deadline for each application, so we do accept monthly. And each approved proposal will be delegated what we call a SPOC, or a scientific point of contact, who you can ask scientific questions to. We do have 5 scopes. We have 1 Arctica with a K3 and fringe-free, a Krios with Falcon 3, K3 and fringe-free, and then 2 Krioses with Falcon 3, BioQuantum, and then 1 Krios with Falcon 4, BioContinuum, K3, and fringe-free. So for sample preparation, we do have a VitraBot and Leica GP2. We are still unfortunately undergoing validation on the VitraJet before we offer that to users. We finally can have visitors on site in March, and we're planning to start our in-person workshop starting in April with sample preparation, followed by a microscope operation in the summer. So look for more information for that on our website and apply today. Ed?
Hi, I'm Ed, coming from NCCAT. It was great seeing a lot of you in the participants list at BPS in San Francisco this past week. If you missed us, we welcome you to come on site for our either access to one of our 4 dedicated Krios instruments, chameleon access, as well as our grid preparation and screening program, which allows you plunge freezing access as well as access to our glaciers, and as well as our embedded cross-training program. A couple of highlights for the cross-training program. In a couple of weeks in March, we'll have an on-site 1-week single particle workshop. That'll be very immersive from end to end for the full Cryo-EM workflow. And all 3 centers have come out with Cryo-EM merit badges. We released the website this past week. And all 3 national centers, once you earn a merit badge, that proficiency merit badge allows you to be cross-honored on another center. So if you want to find out more, go to nccat.nysbc.org, and I hope to see you in New York soon.
Awesome. And then I'm going to stop my share. And then Dr. Kellogg, if you want to start yours.
All right. 1 second. See it?
And then perfect, we do. And then Eugene, if you want to formally introduce her.
Right. Thank you, Lauren. Well, it's my pleasure today to welcome and introduce our speaker, Professor Dr. Liz Kellogg. Liz is currently an assistant professor at Cornell University. Her lab uses Cryo-EM, atomic modeling, and protein biochemistry to harness the power of transposons to engineer novel genome editing tools. Liz will tell us more about her exciting discoveries today. Before her time at Cornell, Liz was a postdoctoral fellow with Eva Nogales at UC Berkeley, where she studied microtubules by using Cryo-EM. One step before that, Liz did a PhD with Dr. David Baker at the University of Washington, where she developed computational programs to model protein-free energy landscapes. For the members of our audience who are Cryo-EM practitioners, Liz published a paper with Mike Cianfrocco in 2020 called What Could Go Wrong? A Practical Guide to Single Particle Cryo-EM. It's actually a really useful guidebook, if you like, for navigating Cryo-EM sample preparation and data processing. So I encourage you guys to check it out. One fun fact about Liz is that she has 2 cats, and one of them is named after Charles Darwin. So today, Liz will talk to us about her work on CRISPR-associated transposons. Liz, please take us away.
Okay, thank you so much. And thanks for the invitation. I'm really happy to give this talk. I'm also happy to participate in the network of national centers. This has been a very instrumental resource for our group. And so, we're very thankful to be a part of that. Okay, so, and what we really focus on are things called transposons in the lab. And in particular, we're interested in transposition mechanisms. And the title of my talk, I changed it slightly from what was posted, but I wanted to focus more on the methods and the methodology, what we use to actually get the structures that we then can learn really cool biology from. So the title of my talk is Cryo-EM Analysis of Dynamic and Heterogeneous Complexes, a case study of CRISPR-associated transposons. And so I'm sure that, some of you may know, but some of you probably don't know what CRISPR-associated transposons are. We usually abbreviate them as Cas elements, so I'll be referring to it as Cas throughout my talk. And I'm sure all of you know what CRISPR is, right? So CRISPR is a bacterial immune surveillance system that everybody's been very excited about because of its ability to revolutionize genome editing in biology. And maybe one of the most well-known examples of this is Cas9, and given a guide RNA, it will seek out the target DNA and make a corresponding cut at that location. And it's been really, really fantastic for generating knockouts, but in order to introduce new DNA or new non-homologous chunks of DNA, you need to invoke the host DNA repair processes. And so that can be laborious, time-consuming, and difficult to do, because that will depend on the cell type that you're actually working in. And of course, I told you that we work on transposons, and some of you may not know what those are, but transposons are actually these we call them selfish genes. So these selfish genes are able to jump around the genome, they're able to self-replicate, and we think of them as selfish in a sense that their primary goal is to ensure their own survival within the context of the host ecosystem, like this host genome as an ecosystem. And transposons encode proteins that are referred to as the transposase, which are really actually very complicated and intriguing from a biophysical standpoint, because these proteins need to recognize the transposon signature, so they need to recognize specific DNA sequences. They need to pair their donor DNA, make cuts, cut it out of the DNA, and then find another target DNA to then integrate this DNA cargo into. So one protein, but many different functions that have to be very tightly regulated at each stage in order to carry out the reaction, so-called, that they need for survival. But transposons actually have been, a subject of interest from genome editing communities because they don't actually introduce double-strand breaks, at least in the type that we're talking about today, the cut and paste transposons. And so from a gene editing perspective, this has also been of some interest, except you can't typically regulate where transposons insert. They typically are, they just insert into open new chromatins. So wouldn't it be great actually if we could combine the activities of, the CRISPR effectors where they actually recognize a particular DNA sequence given a guide RNA and a transposon, right? And that is actually what we're going to be talking about today, these naturally occurring elements for which they can carry out RNA-guided DNA insertions, which means that, they're essentially directed to insert their DNA cargo to a particular location. So we became very fascinated with these. These are elements that are very recently discovered. And so there's not a lot that's known mechanistically about them. So I'm just going to tell you, there's a lot of parts involved with this transposon. This is actually a very complicated one. It contains numerous genes. And this is the right and the left end of the transposon gene. And within the transposon itself, it encodes a number of different protein coding components, which I indicate with these colored blocks. Okay. It also carries around its CRISPR, its protospacers, which we call attachment sites. So it keeps a list of all the different places that it likes to insert within the genome. But in order to make things a little bit more easy for the audience, I'm actually going to refer to the parts of the transposon in terms of their genes and in terms of an analogy. So here the TNSB is actually the transposase here. So it carries the donor cargo and it actually catalyzes the reaction. So that's the catalytic center of the whole element. And I refer to it as the cargo carrier for this story. And this protein is called Tnsc. And Tnsc I refer to as a matchmaker because it turns out that one of the fundamental roles of this Tnsc protein is to actually to find target sites and to recruit the cargo carrier to that target site. And so I think of it as a matchmaker that tries to find matches between potential target sites and the cargo carrying transposase. Okay, so then we have this TNIQ protein, and TNIQ I think of as an adapter. So that's why I have this icon here. I think of it as linking the CRISPR effector components to the core transposition machinery. And then finally, we have this gene called Cas12k. Cas, you can guess that this is the CRISPR effector gene. And indeed, this is actually the RNA binding component of this system, and I refer to it as a target site marker because its function is to bind RNA and recognize the corresponding target DNA location to serve as the target site marker that would then recruit the rest of these components. Okay, so all of these components are required for transposition in this element. And because of a number of different reasons, we actually think that all of these components actually interact at the target site. So given all of these coding protein components, we think that all of these components associate in a manner something like this. So actually the marker, the Cas12k marker, most likely marks the site of the transposon insertion in a guide RNA dependent fashion. And we think that this adapter serves as a bridge between this effector and the rest of the transposition machinery. And we think that, as I said, this matchmaker protein kind of mediates between this target site marker and the cargo carrying transposase. But remember, this is not a CRISPR component. So this does not function in terms of like cutting DNA. It's not a nuclease, and it doesn't function in bacterial immune surveillance. So this is something that actually, this element is actually trying to subvert the bacterial immune system. And so they need to escape detection from bacterial surveillance complexes. And this TNSB, remember, is the transposase. So this is actually bringing the cargo carrying the donor DNA for insertion at the target site. And this story, though, is about this matchmaker protein. And the reason we actually got interested in it because, well, it was a very mysterious component of this Cas system. It's not very common that you would see these kinds of proteins associated with transposons. But what we knew about the Tnsc protein is it's a AAA+ regulator. And AAA+ regulators occur throughout all branches of biology. And if you know anything about AAA+ proteins, their function is to bind ATP and hydrolyze it. So we knew that, you know, this protein was really, I mean, it was very interesting in the fact that we didn't really know much of like what it was doing or how its structure contributed to the insertions of these elements. But there was another experiment that we did that really kind of piqued our interest, right? So like I told you, all of these components are required for transposition. And when we take all of the components, but then we simply change to different nucleotides, we saw something very interesting. So what we use here is actually a transposition assay. So an in vitro transposition assay, we put all of these components into a test tube. And then, you know, we actually give it its donor DNA and then a target DNA and then a guide that recognizes a specific location in that target DNA. And then using PCR, we actually just asked whether the insertions were occurring at the site that we chose, that was dictated by the guide RNA sequence. And if the insertion occurred as planned, then we would see a really bright band that for the left and the right end of those insertions. And in the presence of ATP, everything went as planned. We saw a really bright band for the left and the right ends, which indicated that the programmed DNA insertion was occurring as we expect in the presence of ATP. But in the presence of ADP, we saw absolutely nothing. So this indicates like, you know, there's no targeted insertion. In fact, there's no transposition at all. So that indicated to us that, you know, ATP binding is required for targeted insertions to happen. But what was really interesting is what happened when we used AMP-PNP, which is a non-hydrolyzable ATP analog. And instead of seeing nothing, we saw like this broad smear of bands, which indicated that insertions were happening, but in an unregulated fashion. So the whole programmable insertion was completely deregulated. And so what that told us was not only ATP was required for transposition to occur, but ATP hydrolysis is required for appropriate target site selection, for identification of the right target site. And so what this indicated to us was that this Tnsc protein, or this matchmaker, was playing a fundamental role in the whole process of directing insertions to the right target location. So of course, as you know, because you're at this webinar, you know that I focus on Cryo-EM. And today I'm actually going to be going through a lot of the methodological details that we, and the challenges that we encountered throughout our structural studies, because that actually did not make it into the paper we eventually published. And of course, since you know, this is a Cryo-EM micrograph, and this is actually an image of the Tnsc, or the matchmaker protein, in the presence of DNA and ATP. And what you can see are these beautiful filamentous structures. And so we knew, like, it looked like they were forming these oligomeric structures that didn't seem to be very defined in terms of their length. In fact, any sort of piece of DNA we gave it, this Tnsc protein would actually polymerize along the full length of that DNA. And I just want to actually highlight the scientists who actually did the work here. This study was really led by Jung-Un Park, who was a graduate student in my lab. And there are there's two other trainees in my lab, Amy Tsai and Ishan Malhotra, who also contributed significantly towards these studies. And I'll be actually highlighting their contributions throughout this talk. But we were very, very, very excited about this. We were like, wow, this is so cool, it's so beautiful, right? And of course, as you all know, the first thing you do when you get a Cryo-EM data set is you make 2D class averages. So we actually did 2D class averages. Here's a set of the 2D class averages we got from this sample. And from about 150,000 particles, we get some really nice 2D classes. You can see that, you can see that these layers here, and then you can also see structural features within the 2D classes, which indicated that there was high resolution information there. So we were thinking we were going to get a structure from this. But when we tried ab initio reconstruction using C1, which is no symmetry at all, we ended up with something like this, something that didn't have high resolution features. So you can see that this is this doesn't have any sort of secondary structure information. The resolution of the reconstruction was estimated to be anywhere between 4 and 7 angstroms, but you can clearly see that there's no high resolution features here. And so ab initio reconstructions were completely failing for this helical filament. And the reason we knew it was a helical filament was we actually employed some helical analysis that I learned from my time in the Nagala's lab, which is called layer line analysis. So what we did is you can actually take raw images, and this is actually code that we wrote in MATLAB. And you can actually, so the reason, this is actually a segment of one Cryo-EM micrograph. And in order to do layer line analysis properly, you don't actually need to average your images together. You can just take a raw image and you'll be able to see the layer lines very clearly. But you need to have a lot of filaments in your image in order for this to work. And so this image is the reason I selected this window, is because there's lots of overlapping filaments here, which gives us tons of signal with which to see a layer line. And so this is the real space image. And as you know, being Cryo-EM microscopists, if we take the Fourier transform and look at the spectrum, you'll see very nice tone rings here from that image. And this is actually the Fourier transform that corresponds to that image. And here you can actually see that there's a little ring here. And that's actually the layer lines from all of those helical filaments that are rotationally averaged. So here you can see signal from all the different layers of your particle. This only works for helical filaments, contributing to signal in Fourier space. We do a rotational average of this Fourier transform, and you can see all of these peaks and valleys here. And these peaks and valleys can correspond to the signal in your Fourier transform. So you're going to see oscillations due to your contrast transfer function, but you're also going to see a peak that corresponds to the spacing of your layers in your helical filament. And so this is in reciprocal space, but if we do the conversion, we can then learn, directly from the raw image that our layer line spacing between the layers in our helical filament is 41 angstroms. So that's really important information, right? Because that allows us to then say, okay, we know that these layers are separated by 41 angstroms. It is a very sensitive metric. And so you can actually, back in the day when I was studying microtubules, you can detect spacing differences of anywhere between 1 and 3 angstroms difference in your layer line spacing. And so this can be a very sensitive metric to distinguish how your helical parameters are changing with respect to different conditions. But there's another key piece of evidence that was important here, because as I told you, the ab initio reconstruction was failing. And so by utilizing this information, we could see, like, from the raw images, our helical spacing was 41 angstroms. And another key observation was that all of these layers are stacked on top of each other very nicely, which indicates that there's no helical super twist. Okay, so super twist refers to the fact that the subunits will not actually line up with respect to one another in adjacent layers. And because of that, we could actually guess what the helical parameters were in order to get a suitable initial model. So Jung-Un actually then used RelyOn to generate, I should also mention, all of these details are actually explained in the supplemental data, the supplemental materials of the science paper that we eventually published. You can look it up and read more about it, but also we've posted all of the scripts that you would need in order to do all of these kinds of analyses on our GitHub repo. We have a GitHub for the Kellogg lab. And but in any case, so after doing this analysis, then Jung-Un actually realized he could guess at an initial reference that would then correctly identify the helical parameters, and then also, allow us to get an initial reconstruction. So using utilities in the Rely-On toolbox, he actually then generated 3 different initial models, one containing 4 subunits per turn. So those subunits had a rise that was dictated by that layer line spacing, but with no super twist. One that had 5 subunits per turn, and one that had 6 subunits per turn. And it turns out that we were able to get successful initial reconstructions by using the 6 subunit per turn initial model. So this is the one that eventually worked. And from that, we were able to get this reconstruction here. So that was the key to allow us to get a correct initial model, to allow us to refine the data. And that's what results in what you see here. And you can see this is a top-down view of the filament. And you can see that there's 6 subunits here. And then there's also density in the middle, which I'm going to get to in a second, but that's the DNA that's in the core of this AAA+ regulator. And so after getting this, we were really, really excited, but we actually encountered another problem. So here, what I'm showing is the final structure that we obtained. This is one subunit of the Tnsc protein that's docked into that ATP Cryo-EM map, the one that we actually obtained. And now, so remember, the resolution that we obtained for this reconstruction was estimated to be around 4 angstroms. And thank you so much for highlighting the opinion article that I wrote with Mike Chinfroco, because I actually cite it here. And the reason I cite it here is because we actually went through a lot of the, we give a guide for how you can know whether your sample is working, whether you can build into your reconstruction. It gives you kind of like a roadmap to know whether things are going to work for you or not. And one thing that was really kind of problematic at this stage was the fact that we were seeing these connections here. So even though the reconstruction was estimated to be around 4 angstroms resolution, and remember, we didn't have an atomic model at this stage, and this was before AlphaFold. So we didn't have a guess that would allow us to say, oh, okay, this is probably what it looks like. We just dock it in. No, we didn't have an initial model. So we first started tracing the map, but then really we couldn't get a correct polypeptide trace despite the estimated resolution, where it should have worked. And the reason why is because we were seeing all of these connections here. And you can see that actually these are not due to the main chain. This is due to side chain density that's kind of bleeding through. And so because of this, we were unable to get a correct polypeptide trace at this stage. And if you want to learn more about how, how you would know, how you can recognize this, then read this guide. So this is actually in the ATP bound state. And so we actually weren't able to get an initial model from this. And so then what Jung-Un did is he actually determined the structure of the Tnsc filament in the presence of a non-hydrolyzable analog. So using this non-hydrolyzable analog, we actually use ATP gamma S in this case. We obtained a 3 angstrom reconstruction, which I'm showing you here. And you can see this is actually the same region that I just showed you in the slide before. And what you can see is that there's really high resolution, very nice high resolution features uniformly throughout this filament, which indicates that, which is sufficient for us to build a model. So, so with this in hand, we actually proceeded with model building, and we found that, this is actually the model that we came up with. There's, of course, as I told you, there's 6 subunits arranged in a hexameric fashion continuously, continuous helix, and in the pore here is the DNA, right? And what was really interesting is that we actually learned immediately why ATP was required for initiating transposition in the first place. It was because ATP actually sits between these different subunits here. And so because it's the glue that holds together all the subunits around this helical filament, this helical filament likely does not form in the absence of ATP. And here's the density for the nucleotide that just convinces you that we see clear density for this nucleotide. And this is actually like, this describes the interactions between the different subunits, which we can see very clearly due to the high quality of this map. But another thing that was very interesting about this structure in the first place was the fact was how it was binding DNA actually. And what we could see is that each of these subunits would make a ladder of interactions around the DNA helix. And I actually want to just take a little bit of a tangent here and also explain like how Cryo-EM can inform, hypotheses you have. So for example, each of these Tnsc subunits are colored in a different color, and you can see that it forms hydrogen bonding interactions with the sugar phosphate backbone here, but also you can see that it's all associated with a single strand of the DNA. So it's not actually binding the opposite strand. And if we actually compare this to B-form DNA, in blue is the experimentally observed structure, and in white is B-form DNA. And actually, we see that the DNA is actually lengthened with respect to B-form DNA. So DNA distortions occur along the length of the DNA helix. And very strikingly, you see that it's lengthened by 40 to 41 angstroms. So what we interpreted from this is that actually, this protein, by forming a helical polymer around DNA, is imposing its own helical symmetry onto the DNA filament. So that was very, very cool. But another thing, we actually learned something else, because what you can do, so Cryo-EM reconstructions, we quote one number that represents the resolution of that reconstruction, but there is usually a range of what we call local resolutions throughout the structure. And when we look at the local resolution of the DNA in this Cryo-EM reconstruction, we see the following. So all of these, the voxels in this density, I've actually segmented out the DNA for the map that I was just showing you, and I'm showing you the local resolution in terms of the color. So if it's a higher local resolution, it's going to be more towards the blue, and if it's a lower local resolution, it's going to be towards red. So, and what you can see actually is one part of the DNA is very high resolution, and the complementing strand is of comparatively lower resolution. In fact, it was really striking because it looked like this other half of the DNA got completely smeared out. And from this, we were actually thinking to ourselves that, what, if this is true, then this protein should be able to form helical filaments on single-stranded DNA as well. And in fact, this was true. So we did a negative stain imaging session where we actually take single-stranded DNA and we added this protein and ATP. And you can see that it's not as robustly forming polymers, but it can actually form these filaments on single-stranded DNA as well. So in this way, paying careful attention to the features of your map allows you to formulate hypotheses about your biological systems that you can then test. But in any case, getting back to the story, right? So remember how I told you Tnsc is a matchmaker. It's like it tries to find the target site and connect that with the cargo carrying transposase. But we think that this first structure explained Tnsc's matchmaking function, because no matter what DNA we gave it, Tnsc would form these helical polymers that actually coat the DNA non-specifically. And it also distorts the DNA. And that to us was very interesting, because what that told us was, actually transposition relies on DNA distortions to drive the reaction forward. And so we thought, okay, if it's distorting the DNA, this could presumably be priming the DNA substrate for the subsequent transposition reaction. I don't have time to explain all of the experiments that went into this, but we also know that the Tnsc filaments polymerize unidirectionally, and that's through some image analysis and Cryo-EM reconstitutions that we did, but also the fact that it can also be explained by other facts that I'll tell you later. In any case, Tnsc, we think here, to summarize this, Tnsc is essentially the matchmaker searching for a target at this stage. And so So that's the part of the story that explains its matchmaking function, right? But as you know, this is not, the end of the story. In fact, it's only the beginning of the story, right? Because how does this Cupid protein, how does matchmaker find its target? So we wanted to know next how the landmark Cas12 actually defines the target site. And, remember I told you that there's a whole number of different components here. And there's a CRISPR effector, but we think that there's actually another protein called TNIQ that actually serves as the connector between this CRISPR effector and the Tnsc filament. And I can't go into all the reasons why, but there are other structures out there by Sam Sternberg and Israel Fernandez that shows a physical connection between the CRISPR effector and this TNIQ protein. So we thought that, it's probably serving as a connecting point. But we wanted to then visualize what this connection looked like between the TNIQ adapter and the Tnsc protein. So what we did next is something that, so we utilized the fact that actually this TNIQ protein is relatively small. It's only 18 kilodaltons. And because of that, it will appear essentially invisible in an electron microscope, especially compared to the size of the helical filament that is formed by this Tnsc protein. So what we did next is we actually just saturated with excess of TNIQ. And what you can see here is the Tnsc filament very clearly. These are the filaments that are being formed. But any cryo-electron microscopist that's looking at this image will notice that there's a high background in all of these images. You can see that it looks kind of grainy in the background of these images. And that's because of the high excess of this TNIQ protein that we threw into the mix. So the reason we did that was because we wanted to be very, very sure that TNIQ would have all the chances that it could have in order to associate with the polymerizing filament Tnsc. And when Jung-An processed this data, what he saw in the 2D classes is his Tnsc filament, right? So here's the Tnsc very clearly. And what I have here are simulated projections that you can actually use to compare with the experimental class averages that we have here. So you can see, so for example, take these two as an example. You have a simulated projection here, which is the filament. And then you have the filament here, but you also have a little bit of extra density here. And it's very weak, but this represents the capping TNIQ protein. So this arrow is actually pointing to that weak density. Here we have this is the same view that corresponds to this experimental 2D class. And you can see that there's very weak density here that would be capping the filament. And so we knew that this TNIQ protein is there. But it could be substoichiometric. The density is relatively weak compared to the rest of the filament. And so it's either substoichiometric or it's getting averaged out. And so what Jung-An did then is he employed some 3D classification strategies. And the first thing that you need to know about 3D classification is that it's going to try to separate your particles based on the biggest difference between those two particles. So what I mean by that is that 3D sorting just based on the entire filament would then, you see here is the top view, and you can see that TNIQ, you can see extra density that's not associated with that matchmaker protein. This is the TNIQ density, but it doesn't look like anything that is real. And if you look at it from the side, actually, you see that the 3D classification in the absence of any sort of masking strategy tends to separate these Tnsc filaments based on their length and not on any other feature associated with these filaments, because that's the biggest difference that separates these two classes of particles. So that's the first thing that Jung-An did. But he also could then do focus classification by masking out a single TNIQ. So by masking out a single TNIQ, this is actually in light blue is what he has masked out of the filament. And what we're looking for now in terms of this focus classification strategy is the presence or absence of a single protein here, because we're actually looking for TNSC should be present in all of these cases, but TNIQ would be either there or not, right? So we're looking for differences in terms of 18 kilodaltons of mass. And when we do focus classification based on that, we identify one TNIQ binding here and another TNIQ that is essentially a cleft here between two TNIQ binding spots here. So what that means is that we're actually able to separate out the different TNIQ binding sites on the polymerizing face of this Tnsc filament. And from that classification strategy, Jung-Un then identified two different populations in which TNIQ is occupying the face of this Tnsc filament. So this actually starts to fill in the model that we have so far. We basically think that this Tnsc AAA plus protein is polymerizing in a searching state, looking for its target site. And when it finds its target site, it's actually marked by this Cas12K and TNIQ, and it will form a complex with that adapter protein TNIQ. So that's what we have so far in this story. But so we also wondered, okay, so like now, how does the transposase actually get brought to the target site, right? Because this is actually forming a long helical filament. It's potentially, you know, just as long as the DNA substrate it's polymerizing on. But, you know, based on past literature from mu and the TN7 fields, we actually knew that probably the transposase itself is stimulating nucleotide hydrolysis and filament disassembly. So in the presence of ATP, when we formed these Tnsc filaments, we saw these really nice white particles. This is negative stain. So your particles will appear white on a black background. And when we added the transposase or the cargo carrier, TNSB, we see that the filaments actually disappear. So, you know, but when we actually polymerize TNSC in the presence of AMP, PNP, the addition of the cargo carrier TNSB actually has no effect on the formation of these filaments. So that told us that filament disassembly was nucleotide dependent and also facilitated by TNSB. So in terms of the analogy that we have going on here, the matchmaker protein TNSC is actually, in this case, bringing back, we think, the transposase TNSB to the target site. And remember, just to remind you about how this works, we have this Cas12K protein here, and there's all these other proteins in between, but eventually this transposase gets recruited last and has to be brought to the target site, presumably by this TNSC protein. So at this point, it was, we had kind of a mystery on our hands because the TNSC protein, as I've shown you throughout this entire talk, it forms these continuous helical filaments, right? We've never seen a case in which it hasn't formed this continuous helical filament. And yet, how can it form a continuous helical filament and then also bring this cargo carrier back to the site where Cas12K is binding? There has to be filament disassembly, but we also thought that there must be a form of TNSC that's stabilized that could potentially explain how it might serve as a link at the target site. And I'm going to, so since I'm kind of on the road this week, and so I have to apologize for this is the worst slide in the entire talk, but that's because, okay, I'll just get into it. So what we did next is, you know, we knew that these TNSC proteins were unstable in the presence of ADP. And with in the work with Amy Tsai, we actually did something that, you know, a lot of biochemists and cryo-EM people do. We actually ran size exclusion. So in the presence of these ADP, the TNSC, we could get something that would migrate to a larger molecular weight here, but we could see all sorts of different peaks, which indicated that there was a spread of different sizes, molecular sizes. And this complex, whatever we were trying to reconstitute, was unstable once it was subjected to size exclusion. And if we took one of the peaks that corresponded to the appropriate molecular weight that we were expecting, we would see something like this. And I really apologize for the quality of this image. I don't have API on my screening microscope, and so the screenshot will just have to do. But what you can see, hopefully, from this image is the fact that there's not very homogeneous sizes and shapes of this protein. In fact, it actually looks really like a lot like aggregates here. But when we actually take we take the complex from before sizing and a very concentrated complex sample, we actually see something that looks like this. So we could see from the negative stain that it was forming some sort of assembly that looks regular enough to be subjected to imaging. And rather than trying to, stabilize it upon dilution, we simply imaged it. So this is a Cryo-EM micrograph. That's an example of one of the samples that we imaged using this reconstitution, not with ADP, but with a nucleotide analog called ADP-ALF3. And what you can appreciate from this image is that it's also very noisy. So it has a very high background. It's likely because there's a substantial number of monomers of Tnsc in solution here. So not only did we have to go to very high concentrations, but also this is not a very stable assembly and it tends to fall apart upon dilution, as you just saw. So this image actually demonstrates very well that, this has a very high background. It's relatively difficult to see your particles, but based on the pre-processing strategies we utilized in the lab, we could see that in fact, we were actually getting very beautiful 2D classes from these images. And so I think that, pre-processing is a very important step that can't be overlooked in data acquisition. And it's very, very important to monitor the quality of your data. But hopefully you can also appreciate something else from these 2D classes. These are not continuous helical filaments anymore. And when we do the Cryo-EM reconstruction, we see that what we see, in fact, is actually 2 hexamers of Tnsc. And we think that the reason they're actually oriented in opposite directions is because a stabilizing factor, presumably the TNIQ adapter protein, would then sit at this other interface and stabilize this hexamer against full disassembly. So that rounds out the model of how we actually put together everything in this story. We think this matchmaker protein Tnsc polymerizes along DNA looking for its target site. And once it encounters this target site, it is stopped from polymerizing. It recruits the transposase via nucleotide hydrolysis and filament disassembly, but we think the post-hydrolytic state is actually a stable form that would be stabilized in the context of this complex at the target site. So with that, I actually come to the end of my presentation. I want to make a plug for ACA and M&M, so American Crystallographic Association and Microscopy Society of America, are both in Portland this year, and they're overlapping in terms of the date. And we actually we're getting together these joint events for both meetings. And so I think that, this is a really ripe opportunity for the entire Cryo-EM community to get together in one place. And we have some special scheduling for this particular event. And their abstract submission is still up. M&M's abstract submission is today at midnight. So please get your abstract in. Finally, sorry, one more thing. I'd like to make a plug for my lab. We're always looking for talented graduate students and postdocs. And we just got a new cryo screening microscope. So we have an F200 CI that's just steps away from the lab, and it's going to be a fantastic pipeline for Cryo-EM sample prep and structure determination. So if you're interested, reach out to me. And thank you to everyone who did the work in my lab. So. I'm done.
Awesome. Thank you, Liz, for such a great talk. We do have a couple Q&As. So if you're ready, I can get you started off in that.
Share? Maybe
yeah, let's take it down.
Okay.
All right. So
happy to answer questions.
The first question is, how did you generate the synthetic 2D images that was used to compare against the 2D class averages?
Oh, the synthetic. So there's a utility in Relion called Relion Project. And so you can actually just take a 3D reconstruction and it will project it in all sorts of different views. Yeah, that's very useful for comparing experimental 2D class averages against simulated projections.
Perfect. And then the next question we have is, so, hi Liz, it's nice to see that you have negative stain images. I'm curious, what kind of microscope do you use for negative staining? And then do you typically test samples in negative stain before cryo? Is it worth it? I've heard some argue that high resolution IM, it's not worth testing with negative stain and they just go straight to cryo.
Hi, David. David Durano and I used to be friends. We are friends, but we were in the same graduate school. We were at UW together, so it's really nice to see you here. Right, for negative staining, we used amorgagni, and that unfortunately is out of service now. But we are probably going to use this new F200CI for negative stain imaging. It's interesting to hear you say that some people argue that it's not worth it. I think that it's actually an indispensable step of structure determination. And there's many things that you can learn from negative staining that you can't, that would be difficult to get like a very quick, easy answer in cryo. So for me, I actually require that all my students show me a good negative stain image before they go to cryo. I think it's absolutely worth it. And even like negative stain data acquisition can tell you a lot from the 2D classification.
Awesome. Do any of the other Cryo-EM centers want to weigh in on that as well?
Yeah, please.
Ed, Mike, no?
No?
All right. So another question that we had is, the filaments in the micrographs appear to have different lengths. Is there any significance to that?
So I don't know. I don't know what that question, if that question has a deeper interpretation to it, because so we give our substrate, our substrates are a defined length when we do the reconstitution. And in fact, that's a good observation, because what you see very routinely is that, you see actually the filaments are much longer than what the substrate would dictate, even if we account for the fact of the DNA distortion. So what I interpret it as is end-to-end joining of the filaments. But whether there's a biological rationale for this, I'm not actually quite sure yet. It might just be that because our reconstitutions are in such high concentrations, they form more robustly than they would otherwise.
Awesome. Another question is, do you have any questions with orientation bias in your helical filaments?
So that's one of the best things about helical reconstruction. You never have an orientation bias, because usually you get your side views of your helical filament, and because of the helical symmetry, you actually fill out Fourier space relatively uniformly. So that's never an issue with helical filaments. It's one of the nice things about helical filaments.
That's awesome.
There's a lot of other problems that are like in helical reconstructions, but that's not one of them. So
I had another question is, when doing mass classifications of TN1Q, do you need to use some sort of special processing tricks or parameters to deal with the low signal to noise ratio? For
processing TNIQ?
Yes.
So I believe that in order to do the kinds of focus classification that Jungin was doing, we had to alter the tau parameter in RelyOn. So this tau parameter actually weights the amount of prior information versus experimental information. But other than that, I think other than that, we didn't really have to do anything very special, except for paying attention to the different features that were getting sorted out, and paying attention to our image processing pipeline very carefully.
Another question is, so great talk. How does TN1Q connect the core transposition machinery with Cas12K? Is there evidence for a direct interaction between the TN1Q or the TN1Q Tnsc complex with Cas12K?
Yeah, I mean, I think that that's actually, something that we're trying to figure out right now. So I think that there's other, models for how TNIQ functions in this element. I think that without getting into too many details, I can explain our rationale for this. I think that, TNIQ actually is related to these target site selection proteins in these TN7 elements that are referred to as Tnsc. And Alba Garnet is here, who has done some really spectacular work on the Tnsc from TN7. In any case, the TNSD, TNSD proteins actually are DNA specific binding proteins that actually mark the target site, which TNIQ is homologous to. And so the function of this protein is relatively conserved throughout many of these, throughout the TN7 elements and now these cast elements here. And so what we think is that its function is probably conserved. And if that's the case, it's probably associated with the target site in some fashion. And then, as I said before, there is a direct physical connection between the TNIQ and the cascade effector in the 1F3 system that was solved by Sam Sternberg and Israel Fernandez. So that's the evidence that suggests that TNIQ is associated with the CRISPR effector at the target site. The other thing is that, as I said, TNIQ is essential for transposition. And so probably, it's playing an essential role in target site selection here. So all of that kinds of points to our model supporting the idea that TNIQ is interacting with the target, with the effector at the target site. But I have to say, it's like, there's no evidence, no direct evidence for that right now.
Another question, have you ever processed your data through CryoSpark?
Yeah, so actually we have a pipeline that I can briefly elaborate on now. So first, what we do is during data collection, we actually pre-process using Warp, and our collection is through CRISPR-EM. And then from Warp, we actually then go through CryoSpark in order to get 2D classes and hopefully a 3D reconstruction. And that's important for us, as I said, to monitor how data acquisition is going. And from there, we actually import that or export that to Relion. So that's our typical processing workflow.
We have a follow-up to that question. So more broadly, for someone who's new to Cryo-EM, would you recommend that they learn CryoSpark or Relion first?
I think what I can say is that no matter what method you use, be sure that you understand what parameters you're setting and how to, and the theory behind whatever you're using, because that is going to inform, it's going to allow you to do things that are, it will allow you to formulate your own strategies to tackle whatever problems you're seeing. I think that both CryoSpark and Relion are very, very good programs, and they're very good for different things. And I can elaborate on that if anybody is curious.
We have another question. Can you get the ab initio, I'm sure I'm saying that right, ab initio model right if using very tight circular mask after recentering the particles?
Hi, Min, it's nice to see you. So I don't, I don't know that we ever tried that, but I mean, we tried very, very hard to get an ab initio reconstruction from those helical filaments. And the only thing that really worked was guessing the right helical parameters to begin with. So I don't know that if we tried that specifically. I don't know if we tried that specifically. I'll have to check and get back to you.
Looks like the last question we have, which is good because we're almost to our hour. So I just want to thank you for that great talk again, Dr. Kellogg. And then thank you all, everyone that is with us in this webinar, for joining us. We do wish you well in your Cryo-EM endeavors, and we as National Centers are here to support your science. So let us know if we can help you, and we hope that you join us again in March. Eugene, if you want to go ahead and stop the recording.